Hi everyone. Today in this video we are going to discuss eukaryotic transcription. Eukaryotic transcription, it is a process by which the information that present in the DNA is copied into the new molecule of RNA, and it is the first step of gene expression. We all know the central dogma of molecular biology. That means DNA to RNA through the process of transcription, then RNA to mRNA or protein, it is by the process called translation, then again to DNA. This is called as central dogma of molecular biology. So the gene expression, the first step is transcription, in which a particular segment of DNA is copied into RNA, especially mRNA, by the help of an enzyme called RNA polymerase. And it will result in a complementary anti-parallel RNA strand, which is called as primary transcript. This is the introduction to the transcription, which is taking place in eukaryotes. As simple as that, it is a process by which the information from the DNA copied to the RNA molecule, and that molecule formed from DNA is called as primary transcript. For this process to take place, an enzyme is required, that is RNA polymerase. In eukaryotes, the process is similar to that of prokaryotes, but there are some major differences between the eukaryotic transcription as well as a prokaryotic transcription. In case of eukaryotes, we know eukaryotes means it's complex than the prokaryotes. So the initiation step, which in the transcription process, it is usually complex than prokaryotes. Then the termination phase of the transcription, it also does not involve the stem loop structures. And the main important point to remember is that in eukaryotic transcription, three enzymes are involved: RNA polymerase 1, RNA polymerase 2, and 3. In prokaryotes, only one enzyme is involved, but in case of eukaryotic transcription, there are three enzyme involvement. Then the regulation of transcription is more extensive than that of prokaryotes. This is the major differences between eukaryotic and prokaryotic transcription. So, enzyme, we can look what are the functions and where those enzymes are located. Unlike the prokaryotes, where all RNA is synthesized by a single RNA polymerase, the nucleus of the eukaryotic cell has three RNA polymerase, which is responsible for transcribing different types of RNA. We already learned that prokaryotes contain only one RNA polymerase, single RNA polymerase. But in case of eukaryotes, the nucleus contains three RNA polymerases, which is responsible for transcribing different types of RNA, tRNA, rRNA, small nuclear RNA, etc. So, what is the function of RNA polymerase 1? It will transcribe the 28S, 18S, 5.8S rRNA genes. We all know the subunits of rRNA. This will be transcribed by the help of RNA polymerase 1, and it is located in nucleolus. What is nucleolus? It is the largest structure in nucleus of the eukaryotic cells. Okay, the largest structure in nucleus of eukaryotic cells is called as nucleolus, where the RNA polymerase 1 is located. What about the second enzyme, RNA polymerase 2? It will transcribe the protein coding genes to yield pre-mRNA and also the genes encoding small nuclear RNA involved in rRNA processing, then small nu- um, small nuclear RNA involved in mRNA processing. Then this is located in nucleoplasm. What is nucleoplasm? Which is present inside the nuclear membrane. Nucleoplasm means which is present inside the nuclear membrane to store certain DNAs. There will be RNA polymerase located. 2. RNA polymerase 2. Then, what is the function of RNA polymerase 3? It is also located in nucleoplasm and it will transcribe the genes for tRNA, 5S rRNA, U6 snRNA, 7S RNA, which is associated with signal recognition particle involved in translocation of proteins across the endoplasmic reticulum membrane. This is a function of RNA polymerase 3. Then each of these 3 eukaryotic polymerases contains 12 or more subunits. Another important point, these RNA polymerases which involved in transcription contain 12 or more subunits. So they are large complex enzymes. The genes encoding some of the subunits of each eukaryotic enzyme show some similarities on basis of their DNA sequence to that of E. coli RNA polymerase. However, 4 to 7 other subunits of each eukaryotic RNA polymerase are unique in that they show no similarity with this bacterial RNA polymerase. So these are the basic things about the enzyme involvement and their location, their function, etc. Next, what are the features of eukaryotic transcription? Transcription in eukaryotes take place within nucleus, we all know. An mRNA moves out of the nucleus into cytoplasm. For what? For translation process. So, to translation to take place or the gene expression to take place, first transcription should occur. Then this transcription process, all the process of the transcription will take place in the nucleus, and after the formation of the mRNA or primary transcript of pre-mRNA, it will move out of the nucleus to the cytoplasm for the translocation process. Translation process. It will translocate from nucleus to cytoplasm for translation process. The initiation of RNA synthesis by RNA polymerase is directed by the process of presence of promoter site. Where is the promoter site present? In the 5' side of transcriptional start site. We know the direction of the synthesis is 5' to 3'. Then the 5' on the transcriptional start site, on the 5' side, promoter site will be present. Then the RNA synthesis by the help of this RNA polymerase is directed by the presence of this promoter site. RNA polymerase will transcribe one strand, the antisense strand of DNA template. What is antisense strand of DNA template? It is a non-coding DNA strand of a gene that used as a template for producing the RNA. That is the antisense strand of DNA template. RNA synthesis does not require a primer. Primer means a short single-stranded nucleic acid. This RNA synthesis does not require that. Then, we told that RNA synthesis will occur in 5' to 3' direction with RNA polymerase by nucleophilic attack on the 3' OH, that is hydroxyl group, which is necessary in synthesis of new nucleic acid molecules of the RNA growing chain. And on the incoming ribonucleoside 5-triphosphate. mRNA in eukaryotes is processed from primary RNA transcript. From DNA, the information copied to the RNA, that is called the primary RNA transcript. Then the mRNA become mature from this primary RNA transcript. That process by which this mRNA get matured from this primary RNA transcript is called maturation. These are the features of eukaryotic transcription. I hope you understand. You should keep in mind this point is very important. Where does this transcription take place? What all enzymes are involved? And in which direction the RNA synthesis take place? And what all things or what all steps are involved, etc. This is the detailed process of eukaryotic transcription. This mechanism is divided into different phases: initiation phase, elongation phase, termination phase. First phase is initiation phase. What do you mean by initiation phase, children? During initiation, RNA polymerase will recognize a specific site on the DNA. It will move through the DNA and it will suddenly recognize a specific site on DNA where the upstream from the gene that will be transcribed, called a promoter site. We told that the promoter site will be present on the upstream of the transcriptional start site, which will direct this RNA polymerase. And once this RNA polymerase recognize the specific site, it will start unwinding the DNA locally. Most promoter sites for RNA polymerase 2 include a highly conserved sequence. Most promoter sites of what? Of this RNA polymerase 2 enzyme, they contain a conserved sequence which is located on 25 to 35 base pair upstream. Where this upstream located? Towards the 5' side of the start site, which has a consensus TA, that is Tata box. See, here it is clear on the blue image. Upstream means towards a 5' end. Downstream means towards a 3' end. So, towards this upstream end, in the lower image you can see Tata box is located on the 25 to 35 base pair upstream side. Since the start size is denoted as position +1, see in this image you can see at the right side +1 transcription start site. Tata box position is located at -25. -25 Tata box will be present in eukaryotes, and in prokaryotes it resembles in -10 sequence. This is another major difference. RNA polymerase 2, it contains a Tata box, highly conserved sequence Tata box. In the promoter site, that is on the -25 position in eukaryotes and -10 position in prokaryotes. Both these elements have the same function, namely the recognition of RNA polymerase in order to position the enzyme at the correct location to initiate the transcription process. To initiate the transcription process, this enzyme should locate at a specific position. The sequence present around the Tata box is very important because they influence the efficiency of the initiation. And transcription is also regulated by upstream control element that lie 5 prime to the data box. 5 prime means upstream, means it is towards a 5 prime end. So it is regulated, upstream control elements are present and it is regulated. This transcription is regulated by this upstream control elements, and the sequence around the data box also plays an important role because they influence the efficiency of the initiation. Some eukaryotic protein coding genes will lack this data box. Instead, they will have an initiator element that's centered around the transcriptional initiation site. In last slide, we learned about this RNA polymerase will have a highly conserved consensus called data box. But some of the eukaryote protein coding genes will not contain this data box, but instead they will have an initiator element. Okay, so we can look what will RNA polymerase 2 will do. In order to initiate the transcription process, RNA polymerase 2 requires some assistance of several other proteins or protein complexes. These protein complexes are called as transcription factors, which will assemble into a complex in the promoter region in order for RNA polymerase to bind and start the transcription. Okay, this RNA polymerase 2 enzyme needs some assistance of several proteins and complexes, protein complexes called transcription factors. Once this transcription factors assembled into a complex on the promoter site, then this RNA polymerase will bind and start the transcription. This all have the generic name of transcription factor 2 for transcription factor for RNA polymerase 2. Okay, this all have a generic name because for RNA polymerase 2, this transcription factor assistance is needed to start the transcription. So they are called as transcription factor 2. The first event in initiation means it is a binding of this transcription factor 2 D protein complex to the data box. First event of this initiation, this transcription factor 2D protein complex will bind to this data box through one of its subunits called data box binding protein. Through the subunit of data box binding protein, this transcription factor 2D protein complex will bind with the RNA polymerase 2. As soon as the transcription factor 2D complex is bound, another transcription factor, that is A, transcription factor 2A, will bind and stabilize the transcription factor 2D data box interaction. First transcription factor 2D data box interaction will occur, and to stabilize this interaction, data box 2A will bind. Then next, the transcription factor 2B binds to transcription factor 2D. Now it is more complicated, right, because of the involvement of several types of transcription factor 2, but it's very simple when we listen to this carefully. First, the involvement of transcription factor 2D to the data box. To stabilize this interaction, 2A transcription factor 2A will bind. Next, A, after A we can remember B. So after that, transcription factor 2B will bind to this transcription factor 2D. However, the transcription factor 2B can also bind to RNA polymerase 2. So they can act as a bridging protein. An RNA polymerase 2, which has already complex with transcription factor 2F, now will bind. And this will be followed by the involvement of the another transcription factor E and H. This final protein complex will contain at least 40 polypeptides that called transcription initiation complex. So, what all transcription factor we have learned? Transcription factor 2D, then A, B, then after that F, E, H, etc. Those protein coding genes have an initiator element instead of data box. Now it's in the hand of the eukaryotic protein coding genes, which doesn't contain this data box. Instead, they contain an initiator element. We already learned in the first point. What they will do? They also need another protein that binds to the initiator element. Okay, already they don't have data box. Instead, they have an initiator element. This initiator element also need an assistance of another type of proteins. Then other transcription factors then bind to form the transcription initiation complex, which is similar manner which we have already described above for the genes that possess a data box promoter, which is similar. The genes which have the data box promoter also need to form this transcription initiation complex. Then with the genes which lack this data box also need to form this transcription initiation complex by the help of another proteins. These are the differences which have the data box and which lack the data box. I hope you understand. Next phase is elongation phase. After the end of the initiation phase, that is we have already started the initiation of the transcription and we have finished the initiation, then it will enter into the elongation phase. The transcription factor 2H have two function. First function is it is an helicase. Helicase enzyme means it can separate the DNA double helix into single strand, which use the ATP to unwind the DNA, allowing the transcription to begin. This is a first function. And what is the second function? This is very important and interesting, okay? Because in addition, it will phosphorylate the RNA polymerase II. What do you mean by phosphorylation? Addition of phosphate group, which causes this enzyme to change its conformation and dissociate from the initiation complex. We have already seen that in the initiation phase, the RNA polymerase 1 form an transcription initiation complex. RNA polymerase 2, okay, RNA polymerase 2 form an transcription initiation complex. Then when it reaches in the elongation phase, it needs a phosphorylation. Addition of the phosphate group will change its conformation, which help them to dissociate from this complex. Where this phosphorylation will occur? It will occur on the C-terminal tail, which is called as C-terminal domain or carboxy-terminal domain. This carboxy-terminal domain, I hope this have 52 polypeptides, which is essential for its polymerase activity of the RNA polymerase 2 molecule. So, where did this phosphorylation take place? At the C-terminal tail, which is called at CTD, that is carboxy-terminal domain of the RNA polymerase 2. Then another interesting thing. Only RNA polymerase 2 that has a non-phosphorylated CTD can initiate the transcription. Okay. In initiation phase, you can see RNA polymerase 2 doesn't want any phosphorylation to take place to initiate the transcription. But only an RNA polymerase 2 with a phosphorylated CTD can elongate the RNA. This is an important point. Initiation RNA polymerase 2 doesn't need the phosphorylation, but to elongate the RNA, it needs the phosphorylation at CTD, carboxy terminal domain, which can elongate the RNA. Now it will start moving along the DNA template, synthesizing the RNA. That is, the process enters the elongation phase. After the initiation, now the process will enter to the elongation phase. RNA synthesis in first slide in the features of eukaryotes, we have already seen RNA synthesis will take place in the 5 prime to 3 prime direction. RNA molecule made from protein coding gene by RNA polymerase II will be called as primary transcript. Okay, it is called as primary transcript. This is elongation phase. Then another last phase is termination phase. What is termination phase? Elongation of RNA chain continues until the termination occur. Until the termination occur, the elongation will continue as it is. Unlike RNA polymerase in prokaryotes, RNA polymerase II does not terminate the transcription. Another important point, this RNA polymerase II does not terminate the transcription at specific site, but rather it can stop at varying distances in the downstream of the gene. Downstream means towards its 3' and at the downstream of the gene at varying distances it can stop the transcription. It can stop the transcription, but it cannot terminate the transcription like the prokaryotic RNA polymerase. RNA genes transcribed by RNA polymerase II lack any specific why this termination is not occurring means it lack any specific signals or it doesn't contains a sequence to tell the RNA polymerase II to terminate or to stop the transcription at specific location. To tell that RNA polymerase II, it's enough, stop here, terminate the transcription here. For to tell this, there doesn't contain certain sequences at specific locations and there doesn't contain any signals, specific signals for that. That is the reason why RNA polymerase II in eukaryotes cannot terminate the transcription. So how this termination is occurring in RNA polymerase II? It is cleaved at an internal site. Internal site, the cleavage will occur before it finishes the transcribing. This will release upstream portion of the transcript, which will serve as initial RNA for further processing. Okay, this cleavage site is called as end of the gene. That means it terminates there. The transcript will cleave at the internal site means it is the end of the gene. The transcription process terminate. The remaining portion or the remainder of this transcript will be digested by this exonuclease activity, 5' exonuclease activity. While it is still being transcribed by RNA polymerase I, we told that it cannot terminate. It cannot stop transcribe. It cannot terminate the transcription process. So it will be still being transcribed by this RNA polymerase II. So after the cleavage in the internal site, before RNA polymerase finishing the transcription, it is called as the end of the gene, considered as end of the gene. Remainder will be digested by the exonuclease activity. Then it will catch up to RNA polymerase II by digesting away all the overhanging RNA after the cleavage. The remaining portion is called as overhanging RNAs. It will be digested. It helps disengage of the polymerase from its DNA template, finally terminating the round of transcription. This way, RNA polymerase II is terminating the transcription process. So I hope you understand the termination process. Now after the termination, we have already learned about the initiation phase, elongation phase, termination phase. Now in first slide, in features of eukaryotes, we have learned that the primary transcript or the pre-mRNA should be matured. To be matured, it needs to go through certain processing that is called as RNA processing. We can look what are the RNA processing. The mRNA transcript is much longer localized in nucleus, we told. After the termination process, this mRNA will be moves out of this to the cytoplasm. Okay, it will undergo various steps to change to a mature RNA. It should be matured to become a protein. So it should go through certain steps of process. First one is cleavage. Cleavage means we know large RNA precursors will be cleaved to small RNAs, and primary transcript will be cleaved to ribonuclease pRNA enzyme to form 570 RNA precursors. That is the first step. What is capping and tailing? Children, do you know the structure of mRNA? Remember the structure of mRNA? You can see that initially at the cap position, 5' end of the cap, it will consist of 7-methyl guanosine, and at the tail it can contain poly A tail, that is poly A adenine 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 at the 3' end. It is added. The cap is chemically modified molecule of guanosine triphosphate. Cap means head portion. Head portion contains 7-methyl guanosine initially, and tail will contain poly A tail. We call it as poly A tail mRNA. And cap means it is chemically modified molecule of GTP. Then another is splicing. Third step is splicing. Splicing means this primary mRNA is made up of two types of segments. This is another important thing, that is non-coding regions and coding regions. Non-coding regions are called introns and coding regions are called exons. So we need only coding region. We doesn't need this non-coding region. What is the purpose of non-coding regions? So we need coding regions instead. So non-coding regions are called introns and coding regions are called exons. Then the introns will be removed by RNA splicing, where ATP is used to cut the RNA. By utilizing this ATP, the introns, that is the non-coding regions, will be removed from the RNA. Then releasing the introns and joining exons or ligating these exons or coding regions to produce a mature mRNA. This process is called as RNA splicing. Then another or last RNA processing is nucleotide modifications. They are most common in tRNA methylation, that is then methyl guanosine, etc. examples, and deamination. Adenine from adenine, eosin from adenine, dihydrouracil, pseudouracil, which you will be familiar with the tRNA molecule. Then post-transcription process also required to convert the primary transcript to a functional RNA. So these are the processing, RNA processing to convert this primary transcript into the mature mRNA. This is called as RNA processing. Then significance. Why we need this transcription process? To regulate the gene expression. In first slide itself we have learned the first event in gene expression is the transcription. To gene expression take place, to the gene expression should take place means transcription, the first event should be completed. To further translation should take place for a functional gene expression. For this purpose transcription is needed, and it occurs in preparation for and necessary for protein translation, that we have already told. Translation to take place means transcription should occur first, because it is a first event, and second event is translation. So transcription as the first event of gene expression, it should take place. These are the significance of the transcription, eukaryotic transcription. I hope you understand and like my video. Thank you so much.